Hello, good morning. I hope you guys are doing well. I'm going to go through a few questions in the Lecture 9 study guide. Hopefully none of you have done your quiz yet. And then we will wrap up a few slides in Lecture 9 before we begin with number 10. So here's a gel, and we use a gel in gel electrophoresis. And the purpose of running and doing electrophoresis is to separate DNA based on their size, based on their length. Okay, so there is a gel, and it says we run the gel after plasmid was digested using restriction enzymes. So here it says indicate the following on the gel, where the positive electrode was located. So you should remember when we run a gel, it goes from negative to positive. That's the direction of the electric current. And the reason we do that is because the backbone of DNA is negative. And when you apply a current from negative to positive, you are repelling, you're pushing that negative backbone in the DNA, causing it to move towards the positive side. Now, the gel is a network of mishmash, right? Like a lot of fibers crisscrossing, forming a net, if you would. And so things that are longer, DNA that are longer, they don't really move through the gel as quickly as the shorter DNA. And so each lane here represents a particular DNA sample. And it's asking us to locate the largest band. So in each of the lane, the largest band is always the one that is closest to the well. Okay? These wells, this is where you load the sample. Okay? Samples are loaded here. Okay, and so of all the bands, this one would be the longest. Okay, longest DNA. Because that's the one that is closest to the well, right? In all of the lanes. And the smallest band would be the one that is furthest down, which is right here. That is the one that contains the shortest DNA. Shortest DNA. Okay. Next, we want to know the number of cuts that were made in the plasmid to produce this number of bands. So if you take a look at a plasmid, which is circular, right, circular, and if you cut it one time, like that, then it will just open up and become linear. So that's a single band. So the fact that here in the first one, we saw 4 bands, right? In this first column, first lane, lane A, we have 4 bands, means you actually have to do 4 cuts. So look, if this is the plasmid and I cut it 1 time, 2 times, 3 times, 4 times, then I would have 4 fragments. 1, 2, 3, 4. Right? I can draw it out for you. Right? So you get 4 fragments like that. And when you load that into the gel, it will separate according to the length. The longest one is closest to the top. Right? So for plasmid, this would be 4 cuts. Right? 3 cuts. Essentially, the number of bands that you see on the gel corresponds to the number of cuts you need to make to create those bands. So that's the plasmid. Will the answer be different if linear DNA was used instead? This is linear DNA if it's a straight line. Okay, if the DNA is not in a circle, then how many cuts do you need to create 4 bands? Well, you actually only need 3 cuts. Right? So if you cut it 3 times, then you have 4 bands, right? 1, 2, 3, 4. So 3, 2, 4, 3. Essentially, if it's linear, the number of cuts is one less than the bands. Okay, number of cuts is 1 less than number of bands. Okay, so that's the gel electrophoresis. Okay, moving on. The answer for these are already posted. I just want to go through a few like particularly complicated ones that you might not understand just by looking at the answers. So here we have a DNA fragment, and it's linear. It's not in a circle, and we are going to be cutting it with BAMH. Okay, BAMH. How many DNA fragments would you expect? So here is BAMH, and the recognition site is GGA TCC. Right, so we're treating it with BAMH, so we will look for GGA TCC in this segment. Make sure you pay attention to the direction. So on the 5' to 3' is GGA TTC. So we just look for GGA TTC, which is right here. Right, so that's gonna produce your cut. Okay. Then we keep on scanning through the fragments and see if we have another GGA TTC, and we do not. So that's what happens if you use BAMH on this fragment. You will only cut it once. You have a longer fragment and then a shorter one. If you treat the DNA fragment produced in part A with BCL1, what results are expected? So BCL1 is TGA TCA. Okay, so we will look for TGA TCA, just scanning across, trying to find the same sequence, and there it is, TGA TCA. Okay, so you're gonna have another cut right here. Okay, and that's it. There's no more. So if you use both restriction enzymes, you're gonna have 3 fragments. And now part C, that's the part that requires a little bit of thinking. Okay? So it says we are going to load different DNA samples into a gel, and we have 4 lengths. Okay, so I'm just going to draw 4 lengths here. These are wells, right? To load your DNA sample, they should be the same size, but it's hard to draw on the computer. But this is length 1, 2, 3, and 4. And in each length, we are going to be loading a particular sample. And the question actually tells you what sample we are loading on. So length number 1, we have untreated DNA, just the entire DNA segment without cutting it. So that's very long, right, on its own, compared to the ones that are being cut. And so I would expect to have just a band up here. Okay, the DNA is going to be right here, very close to the top. Okay, very close to the well. The well is where you load the sample because it is uncut. Right? It's very long still. It's not going to get very far. All right. Number 2, DNA sample is treated with BAMH. Okay, so just the blue, right? Just the blue cut, not the red cut. You just have a cut right here. So you have a small fragment and a long fragment. Okay. So the long one is not as long as the uncut, but it's still pretty long. So it's going to be up here, close to the top, but further down compared to the uncut. And this little piece here, it's pretty small, so it's going to go all the way down. Okay? I'm going to highlight this piece here. I'm going to highlight this to show you that they're corresponding to each other. Now I'm going to give you a moment to think about what lane 3 is going to look like. In lane 3, we are going to be cutting with just BCL1. Okay, and I want you to think about what it's going to look like. Okay. So how many bands do you expect to see? How many lines of DNA are you going to see in lane number 3? Anybody? Well, you see there's only one cut, right? With the red here. So you will have 2 fragments. Okay, you will have this piece here and then this whole piece. Right? Remember, we are only treating it with the red restriction enzyme, so the blue cut did not take place. So in this case, we would have the yellow piece, which is quite small, but not as small as the blue one. So I would expect it to be a little bit higher up like that. Okay. And then the other piece that we get, it's longer than the yellow, but definitely shorter than the one we got from BAMH. It's going to be further down. All right. And finally, we have the BAMH1 plus the BCL1 together. Okay. That means we're going to be having the same, we're going to have both cuts here. Right? So you're going to get 3 pieces. Right? You will still get the blue piece and the yellow piece, and then you will have a middle piece, which is shorter than the ones in lane 2 and lane 3. So I will have one right here. Okay, so these yellow pieces are actually identical. That's all. Do we have any questions? Okay. Now I do want to go through this next one with you as well, in terms of like gene insertion. Right. I already told you about the meaning of these things when we did the lesson last week. I'll verbally review it for you, okay? So we have all these things on the outside. Those, again, are the restriction enzyme sites, right? These are locations which you can insert the gene into. And to insert the gene, you have to treat your plasmid and the gene of interest with the same restriction enzymes so that they can create complementary sticky ends, right, to attach to each other, right? So if we want to treat it with EcoR1, then you'll be inserting the gene right here. If you treat the plasmid and the gene with PST1, you'll be inserting the gene right here. So these locations tells you, these markers, I should say, tells you where you can insert your gene. But how do you know whether the bacteria has accepted the plasmid or not? Well, that's why included in the plasmid, we have these antibiotic resistant genes, right? If they have accepted the plasmid and if we have not interrupted the resistant gene, you would be able to grow the bacteria in the presence of the antibiotics. So let's take a look at this question. You inserted the gene by treating your DNA and the plasmid with BAMH and transform it into E. coli. What are the expected results if you grow the transformed E. coli in the following culture? Okay, so we are inserting the gene with BAMH. So we look for BAMH here. That's BAMH. Okay, we're going to be putting a gene right here. Right here. Okay. So if you grow them with no antibiotics, are they going to survive? Well, anytime you grow the bacteria without antibiotics, they will survive. Okay, they will grow. What if you grow the bacteria with tetracycline? Because we have inserted a gene using BAMH1 right here, using BAMH1, we actually interrupted the tetracycline resistant gene, right? Our gene goes right into the middle of the tetracycline gene and destroyed it essentially. So even if the bacteria have accepted the plasmid, they would have no resistance to tetracycline because the gene is not there anymore, right? We interrupted the tetracycline resistant gene with our gene of interest. And the way we did that was because we chose to insert the gene using BAMH. If we insert it elsewhere, it would have been okay. Okay, so this is no growth. No growth. Because the tetracycline resistant gene is disrupted by our gene insertion. Okay, you put something in the middle of it, destroy the resistant gene. Even if they take up the plasmid, they're not going to have resistance for tetracycline. What about growing them in cultures with ampicillin? Are they going to survive? Are they going to grow or not grow? Anybody? We are still inserting with BAMH. Will they grow or not grow? They will grow. That's right. Because the ampicillin resistant gene is intact, right? It has not been disrupted by our insertion. So they will grow. Okay? Ampicillin resistant gene is not disrupted. Okay? Now, very quickly, let's try another one here. If you insert the gene, it's the exact same situation, but this time we use equal R1. So if you use equal R1, the gene's going to go right here, right? Right here. That will not be disrupting the ampicillin. They will not be disrupting the tetracycline resistant gene. So what do you expect? Will they grow with no antibiotic? Grow. Yes. Will they grow in tetracycline? Yes, because they have the resistant gene. And they will also grow with ampicillin. Okay. Grow, grow, grow. No problem. That's all. Any questions? So you will have some of these in the test, right? Multi-select. It will grow in the presence of which of the following? You have to choose all the ones that they will grow in. Okay, last question. Predict what will happen in each of the following situations during gel electrophoresis. So this is your gel. And we're going to have wells again. This is negative. This is positive. Supposed to go this way, right? Predict what will happen If we reversed the electrode, so the current runs from positive to negative, you plugged it in wrong, even though they're color coded, right? Red goes into red, blue, black goes into black, but let's say the person made a mistake, they switched it. So now the current goes this way. What will happen? Well, the DNA is going to get pushed upwards, right? It will exit the gel at the top, right? DNA exits at the top of gel, and is lost. Okay, it's gone. Come out of the gel. What if you run the gel for too long and the dye front is no longer visible? Remember, the dye front is like the blue line, right? That shows the dye, right? And it gives you a rough idea of where the sample might be at, but the dye is gone. It exits at the bottom. Well, what does it mean? Well, if the dye exits, then you can guarantee most of your samples are gone. Okay? Right. Most DNA exits at the bottom. And are lost. Okay, it's gone. Right? Remember, when given sufficient time, even the long ones will be able to make it through. It's completely gone. If you forgot to add ethidium bromide to the staining solution, what happens is DNA will not be visible under UV. You will not see anything. Right? The ethidium bromide is supposed to bind to the DNA and fluoresce, gives off glowing, right? When you shine UV light at it. If you forgot, then you can't see your DNA. Okay? You will think your DNA sample is lost or something. Right? Like it's not, you can't see it. Okay? So there are many things that could go wrong. Okay. So I just want to go back to our PowerPoint here. I just have a few more slides to go through with you. They're pretty simple stuff, but it's a good idea to take a look here. Oops, it's the wrong monitor. There we go. So last time we talked about gene therapy, right? And we have a couple of ways to make this work. Just gonna briefly refresh your memory on what they are. We can replace a non-functional gene. However, this only works, it works best if the gene that is not functioning represent a recessive gene. Okay, so you can replace it with a functional copy. The functional copy, being the dominant one, will override the non-functional gene, and it will supposedly cure the condition. But again, only if the condition have not already created some kind of irreversible damage. Okay. If it has created irreversible damage, then the gene therapy will not fix those damage, but moving forward, it will prevent extra damage to be done. Okay, it will prevent further consequences from the disease. We can also use a gene to suppress overly active gene by inserting a gene that will suppress an oncogene, for example, that could slow the cell division in a tumor. We can modify the clinical course by either adding a suicide gene into say cancer cells, and it will cause them to kill themselves, which is great. The tumor will shrink and it will basically destroy itself. Or we can use a marker gene that causes the Wait, did we do this yet? I actually don't. I might have been confusing you guys with another class. Did I do this already with you guys? Do you guys remember at all?
I don't think so, sir.
You don't think so? Okay. Did we just left off at where did we leave off? Oh no, we did. We actually did. I remember talking about the ex vivo and the in vivo. No, we definitely did, yeah. You guys don't even know, do you? But that's okay. Check the recording. I promise you I did this with you last time, okay? And then the marker gene is gonna cause the cell to put all these signals on the cell membrane, and it will attract the immune cells to come and attack them and destroy them. Okay, so those are the three goals of gene therapy. Okay, we talk about ex vivo, which is doing the modification outside of the body, and in vivo, which is doing the modification, in the body. And now we can talk about some challenges associated with gene therapy. Okay, so they're listed here, and I'm just going to go through the slides with you. Number one, target delivery. How can we ensure that the gene we are delivering goes to where we intended it to go? And let's say we're inserting a suicide gene. How can we make sure it goes into the cancer cells? Okay. If we are inserting a functional gene, how do we make sure the functional gene goes into the intended cells? That's number one. And number two, if it gets into the intended cells, how do we make sure that the gene is going to insert where it's supposed to be inserted into? Okay, so delivering the gene in the wrong cell would be inefficient and could cause other health problems that are unintended. If the suicide gene goes into healthy cells, then we kill healthy cells. That's a bad thing. Even once the right cell has been targeted, the gene has to be turned on. Not only does it have to get turned on, it has to insert itself into the right place. Okay? What if you insert the gene accidentally? Like you have no control, right? Like until we find a better way to deliver these things. Once the gene goes in the cell, what if it insert itself into the middle of another gene? Then you destroy that other gene, right? So that could be problematic. And sometimes cells, they kind of detect these foreign genetic material coming in, and they don't like that, and they could shut down the gene. So even if everything goes well, if you get the gene to the right cell, you park it into the right spot, you might still not be able to turn it on, right? So there's a lot of challenges to overcome. One of the ways we can help with the target delivery is to use a virus. Okay, viruses already have their very specific target. If you think about it, let's say a cold virus, right? A cold virus will infect cells of your throat and in your nasal cavity, right? They are not the flu virus is the cold virus is not going to infect your liver cells, for example. But if you talk about like hepatitis virus, that will only infect your liver, and it will leave all the cells in the body alone, right? So by using a virus, we can actually target where to deliver the gene. If we want the gene to go only to the liver, well, we can learn from the hepatitis virus, right? Of course, we're going to have to take out all the virus DNA that causes the disease, and then we replace it with the gene that we want. Okay, so the virus will deliver the gene to the cell, and it will, insert it into your DNA. But because we're using a virus, your immune system is going to react. And so we can minimize the reaction sometimes, but nevertheless, right, long term, your immune system might be rejecting the gene therapy. So, how do we modify the vector such that we will not be triggering an immune response? That's another challenge. There is also risk that the new gene will insert itself into the path of another gene. I told you that already. It will disrupt its activity. And for example, if you have a gene that is a tumor suppressor gene, the red light that we talk about, right? That slow down cell division. If the gene of interest that we're trying to use in the gene therapy goes into the tumor suppressor gene, then we destroy the tumor suppressor gene. The red light no longer works. Then that could cause uncontrolled cell division, right? So that's what we mean by interaction with other genes. There's so many genes in the cell, it's just hard to predict what's going to happen once you insert something into the cell. So that's why like you can't just do this on humans, right? Like you got to try it out on animals for testing first. Costs. Many of these genetic disorders are kind of rare. I mean, things like sickle cell anemia, beta thalassemia, and hemophilia, the ones that we've talked about previously, those are common, okay? And the cost would be relatively lower. But in many cases, the gene therapy needs to be tailored for individuals who are suffering rare genetic conditions. And for that reason, it is very expensive. There is this guy who was in what is that show called? Like Americans Got Talent or something, I think. I forgot what his name is, but you know, he's a very talented singer that was on the show, but he was born with a form of blindness. And what happens is he after he got the fame right from the show, he was able to gather crowdsourcing funds to get gene therapy to basically fix his blindness. He was born this way, right? So, but then with the gene therapy, he actually was able to see. But the cost is very, very steep, right? The eye treatment costs $850,000, right? Divided by 2, that's like, you know, a little bit under $500,000 per eye. And that's true in general for gene therapy. Right? They ranges from $500,000 to, you know, $1.5 million per treatment. And some therapy requires ongoing costs, right? Another kind of gene therapy for a type of spinal muscular atrophy. Atrophy is like shrinking of the muscle. It causes $250,000 in the first year, and these are US dollars, by the way, followed by $375,000 per year, right? Every year for life, right? So not everybody, let's just say only very few people would be able to afford that kind of treatment. Some company says that they have a pay only if it works approach. If the gene therapy doesn't work as intended, that's okay. You don't have to pay. You don't have to pay for it. But what if it works initially and then it stops working, right? Like, can I recoup my money? Right? That's a little bit of a gray area, right? Some people turn to crowdfunding as a way to get the funds that are needed. This is just another story. It's an old story now about these kids who were diagnosed with a rare form of a mutation in the nervous system, and then the parents cannot pay for it, and so they do crowdfunding, right? And the drug costs more than 1.6 million. Right? In desperate situation, crowdfunding sometimes is the only way forward, and it's a good way. Okay, insurance company often don't pay for these things for, you know, obvious reasons, and that's pretty much it. Okay, for the biotechnology. Anybody have any questions before we move on to number 10, which is the last lecture of the course? Very exciting. Okay, don't forget your quiz is today. It's gonna be 8 and 9, right? Your 2 attempts. Okay? So don't wait until the end of the day. Okay? Do one in the morning, see how you do. If you did great, fantastic. You don't have to worry about it. And if you don't do it as well, you can you know, study a little bit more and then try again in the evening. In the past, people do it one attempt immediately after the other. Okay, that's not the intention, right, of giving you 2 choices. Okay, if you do it and you get like 63 or something, 64, 65, you try it again, most of the time you get the same score. Okay, you got to study a little bit, right, before you can do better. All right, let's do number 10. And lecture 10 is body tissue. And this is a nice connection between this semester and next semester. You will be learning about the body system next semester, where we will take an in-depth look at the body tissues of various organ systems and how they work and what happens when they don't work. Okay, so there we go. Let's take a look. Hold on a second. Let me just see if I can make this a little bit bigger. Nope. All right, so let's talk about stem cells. Stem cells are cells that can divide to produce more stem cells. So they are self-renewing. They could do mitosis to make more copies of themselves. But that's not it. Okay? Stem cells can also differentiate into specialized cell type. Okay? Differentiate is a term that you will hear quite often, as you progress through the course, like next semester. A cell can transform, can differentiate into certain specific cell type. Think of it like us, right? We are all in this pre-health program, okay? But after you graduate from the pre-health program, some of you will specialize in practical nursing. Some of you will specialize in pharmacy technician, right? So that's like differentiating. We go to different paths and we specialize it. The thing about the stem cells, though, is once it specializes, once it differentiates into, let's say, a heart cell, a liver cell, there's no turning back. Okay? They cannot undifferentiate. It's a committed path. Okay, and they stay that way. So that's what differentiation means. There are two types of stem cells. We have embryonic stem cells. Those are the ones that are found in the embryo as the baby is growing. And they are very versatile, meaning they have many options. Okay, they could literally become any cells in the body. Okay. Once the baby is born, though, the stem cells are less versatile. Okay, they are restricted to a particular cell type. Okay, so again, let me use that analogy of going to school again. When you were younger, right? When you are still in elementary school, that's kind of like embryonic stem cells. There's so many options. You can go into business, you can go into arts, you can go into culinary school, so many options. But now when we get older, right? Now you are in the pre-health program, you are more limited now, right? Okay, you're more limited to like the health field, but there are still options in the health field. Yeah, you can do nursing, you can do dentistry, whatever, right? But then it's not like going into finance is not really an option now, right? Now that we're in the pre-health program. So that's what happens. After birth, the stem cells, you as an adult have stem cells in the bone marrow, but those stem cells can only become blood cells. They're not going to become like brain cells, okay? They're limited in their capacity now. But there are different types of blood cells they could become: red cells, white cells, right? Platelets. And then you also have stem cells in your GI tract, in your digestive tract, right? They could become cells of the intestinal tracts, but not blood cell. Okay. And what people try to do is they try to store the embryonic stem cells when the baby is born. They take it from the cord blood, the the umbilical cord, right, that connects the baby and the mother. There's some stem cells in there, and they can freeze it and use it in the future if their child needs it. But of course, there is a expensive fee you have to pay to store them, but it is an option for some people. So here is the stem cell. You have a pool of them, and they can make more just by doing mitosis. Okay, and then when you need to, the stem cells is gonna differentiate. It's gonna become cell type 1, cell type 2, cell type 3. Okay, but remember, this is a one-way path. There's no coming back. Once you commit to that path, that's it. Once you become a blood cell, there's no turning back. Okay. We learn about, we learn about the levels of organization way back in lecture 2, right? We started with the atoms, the molecules, the macromolecules. Okay, we learned the organelles. We learned everything we could about all those cellular processes, DNA replication, protein synthesis, and now we're moving up to learn about tissues. Okay, and as I said, next semester you will learn about organ and organ system. So the whole course, right, in the pre-health basically takes you through this hierarchy, right, until we learn about the entire organism. So most animals, including humans are animals, okay, have 4 types of tissues, and these tissues organize to become the organs in the organ system. So example of tissues include things like fat tissue, muscle tissues, and we'll learn a bunch more throughout the lesson. But in general, these tissues are groups of specialized cells that comes together to perform a specific function. Okay, so we know we have different cell types. And we have cells that are good for storing energy. We have cells that are good in producing movements. So you take a bunch of these cells together, and then they form a tissue. Okay, and then different tissues come together to form the organs, and up we go, the hierarchy. So these are all the body system that you're gonna be learning next semester, except for the skin system. We don't really do that, but everything else you're gonna learn. Okay, you're gonna learn 1 system per week. All right, so 14 weeks in total. If you take away the test 1 and test 2 and whatnot, right? Basically, you have 12 weeks, right? 1 system per week. All right. Many of these organs are going to be found in body cavities. All right. So like if you think about the inside of the body, if you open up the chest, there is the thoracic cavity. Okay, and then further down, like here in the abdomen, you would have the abdominal cavity, and even further down where the reproductive organs, so you have the pelvic cavity, right? So we're gonna label all that stuff in the study guide. Okay, but here it is. This is just to show you where the cavities are. Okay, so cranial cavity is up here. I hope you know the brain is in the cranial cavity, right? Brain. And then there is another vertebral canal that extends all the way down to the bottom. That's where your spinal cord is going to be. The spinal cord runs from the base of the neck all the way to like your lower back. Okay, so I was telling you about the chest. You have your thoracic cavity. So give me 2 things that are in the thoracic cavity. Anybody? You must know, right? What's in your thoracic cavity?
Lung?
Mm-hmm. We have 2 lungs. Okay, they are in the thoracic cavity. Anything else in the thoracic cavity besides the lungs?
Diaphragm?
The heart. Someone said the heart. Sorry, which one is it?
Diaphragm?
The diaphragm? Yeah, is that what you mean?
Yeah.
Yeah. Okay, so the diaphragm is right here. It is not in the thoracic cavity, but it's a barrier that separates the thoracic cavity from the abdominal cavity. Okay, so you have a diaphragm here. That's like a dome-shaped muscle that helps you breathe. Okay, so what about abdominal cavity? What's in there? Give me a look. You should be able to give me a bunch of things that are here. I want 4. Okay, give me 4 things that is in there. Okay, we have the stomach. Good. Is the stomach on the right side or the left side? My class was having a bit of trouble with that as well. Stomach on the right or on the left? Right or left? Stomach.
Left.
The stomach is on the left side. That's right. Very good. Okay. What else? What else do we have in the abdominal cavity besides the stomach? What's on the other side, huh?
Liver.
The liver. Very good. The liver. Okay. Maybe a couple more things. Intestines. Good. Intestines. Just one more. What's in there?
Pancreas.
Pancreas. You guys got it. Very good. Okay. That's the abdominal cavity. Pelvic cavity. What's down there? 2 things. Give me 2 things. You should at least know 2 things that is in the pelvic cavity. Yep, the bladder. Now we want to say the urinary bladder because there are other bladders in the body, like the gallbladder, right? Okay, so that one is urinary bladder. And one more in female, particularly. I mean, you can give me something in men too, but like the female one is probably more well-known, I guess, commonly known. What's in the pelvic cavity for female or male? Ovaries, that's a good one. Yep, ovaries. I was thinking about the uterus, but ovary works as well. Okay, for men, anything? Any men in the class?
Uranus or
Don't-
Uranus. Anus.
Anus is like all the way down here. Okay, that's a good try. Try though.
Testis.
Nope. This is where the penis is, and the testis would be like right down here. Okay, but in the cavity, What do men have to worry about when they are older than 40 years old? Prostate. That's right. The prostate is also in the pelvic cavity. Okay. Anyway, just showing you some of the stuff that are in here. Right on the test, I could ask you like basic things. Okay, I'll be like, which of the following is in the abdominal cavity, and then you should know. I'm not going to give you something that you've never heard of. Okay, it's like obvious things. All right, I'm gonna teach you the anatomical terms, and for this, you would need to have your study guide, which I keep on telling you guys to print. I don't actually know if you guys print it. I hope you do. So there's this picture. Let me see this again. There is this picture that we're gonna fill in together. Okay, fill in together. So label the appropriate anatomical terms. All right, so when we talk about body parts, we need to know where things are. And what's on top and what's below is going to be different if the person is like lying down, or like in different positions, right? So to make it standard, we have what's called the anatomical position. I can't really show you. I guess I can. Like it's just like standing here and then with your palms like slightly apart, just like the picture, right? In the diagram. Okay, that's called the anatomical position. Okay, we'll be doing a lot of labelings in next semester, so make sure you get some colors and stuff like this. Oh, I mean, if you're in my class, we will be doing a lot of coloring and stuff like that. So this is anatomical position. Okay. I am only teaching one second semester section next semester. If you want to be in my class again, it's section 201. If you don't want to be in my class, don't choose 201. Either way, that's information for you to use. All right. So in this anatomical position, we have things that are on top, above, and we have things that are below. Things that are above, we use the term superior. Okay. Superior. Opposite of superior is going to be
anterior.
Anterior is to the front. Good try. Superior and inferior. That's right. Inferior. All right, so let's do some examples here. Complete these sentences using the term superior and inferior. The hands are blank to the feet. Remember, you have to think about the anatomical position, right? Like standing there with the palms facing forward. So what do you think? Is it superior or is it inferior? Your hands are superior, right? It's like should be above your feet. Okay. What about knees and waist? Okay. Waist is here. Knees is this one. Okay. So are your knees superior or inferior to your waist? Inferior is correct. That's right. Do you guys understand? Right? Should be straightforward, right? Elbows is blank to the wrist. Okay. Wrist is here. Elbow is here. But remember, you have to be in anatomical position. So you cannot have your arms up like this, okay? It has to be like the picture that I showed you. So elbows, elbow is it above the waist, the wrist, or below the wrist? It's above, right? All right, the calf is blank to the ankle.
Inferior.
Calf, inferior.
Inferior.
Is it though? Okay, let me show you where the calf is. All right, I can't put my feet down. Calf is right here, okay, back of the knee. And the ankle is, you know, like, I really can't pull my leg that high. Let me draw the leg here. Okay, calf is right here at the back, right? And the ankle is down here. Okay, so calf is superior. All right. I hope that makes sense. All right, ready for the next one? This should be fun. This should be more fun than, you know, I don't know, biotechnology. All right, this is the person looking at it from the side. So we have things that are in front. In front is called anterior. What's the opposite of anterior? What about the back? What's the, anybody know? Opposite of anterior. Yep, posterior. Posterior. All right, now sometimes it's a little bit tricky. Okay? If it's on the side of the tummy, all right, instead of saying anterior, we can also say ventral. Okay, and if it's on the back, we say dorsal. Now, why do we have two sets of terms for the same thing? Well, anterior is not always same as ventral if we are talking about other animals. So let's say we have a dog or something, okay? Okay, the dog. So for the dog, I mean, anterior is still here, right? At the front.
And then posterior is the back. But now the ventral is down. And dorsal is up. So it depends on the position of the organism, right? That's the normal state. That's the anatomical position of a dog, which is different than humans, right? But we also use ventral and dorsal when we talk about things like the spinal cord and stuff like that. All right. So I want you to think about like the shark movies that, you must have seen at least one shark movie in your life, right? And then, you see the fin of the shark. That's the part that is coming out of the water. They usually only show that part because that's like the cheapest part to make, right? Like without building the whole shark, right? For the scene. So which fin is this? Do you guys know based on the terms that we just learned? Well, if you didn't know,
Dorsal.
yeah, the dorsal fin. That's right, the dorsal fin. Okay, very good. Dorsal fin. All right, let's do some more PowerPoints. The heel is blank to the toes. The heel. Okay, so if you think about the feet, heel is right here. Toes, I think you know where the toes are. So this would be posterior. If you think about your arm, bicep is right here. Tricep is right here. So in an anatomical position, your bicep, tricep, what's the right term that goes here? In front of, right? So that's anterior. For the leg, here we go again. The leg here, this is the quadricep. And then the back, you can't really see it, okay? But on the back there, that's the hamstring. Okay. So hamstring is blank to the quadricep. It's in the back, I told you, right? So that's posterior. All right. So that's how you use the terms. Okay, moving on. I forgot to fill this in. This is also superior. If you see A, that means they're the same thing. And then this is inferior at the bottom. So we can divide the person right down the middle. This is called the midline. The midline. Okay. So like the nose is on the midline. The mouth is on the midline. Right? The belly button. Right? Belly button, that's on the midline. We can have things that are further away from the middle, or we can have things that are closer to the middle. Okay, things that are further away from the middle, we say lateral. Things that are closer to the middle, we say medial. Okay. So let's take a look at the examples. The arms, okay? The arms are blank to the midline. Nothing is more middle than the midline, right? So everything relative to the midline, we say lateral. The neck is in the midline. So that would be medial. The shoulders, right, are lateral to the midline. The ears are blank to the nose. Right? Ears are further away from the center, lateral. Can somebody give me another example of two structures relative to each other using these terms? Anybody? Anything at all?
Ebro?
The elbow, okay. The elbow is what?
Ebro.
I'm sorry, which one is it again?
Ebro.
Ebro?
Yeah.
Okay. Okay. Oh, eyebrow?
Yeah, eyebrow.
Yeah, eyebrow. Is what? Give me like you have to have something else, right?
Eyebrows are all there to noise, maybe.
Yep. So the eyebrows are lateral to the nose. Right? Very good. The eyes are medial to the ears, right? Okay. All right. Next is proximal and distal. We use proximal and distal when we talk about things that are on the arm, on the hand, on the whole arm, or things that are on the entire leg. Okay. If it's closer to the shoulder, and we say proximal. If it's further away from the shoulder, then we say distal. For the leg, if it's closer to the hip, okay, that's where the leg joins the body, right? We also say proximal, and then we say distal. Okay. So the fingers are blank to the elbows. Fingers are here. It's further down the hand, the arm, sorry, further down the arm than the elbow. So we say distal. The knees are blank to the feet. The knees are closer to the hip, right? Closer to your hip. So we say proximal. Wrists and fingers. Wrist is closer to the elbow than the fingers are. We also say proximal. That's all. Okay, I have two additional terms. It's not on the diagram, but I have to show you. Ipsilateral means is on the same side of the body. As in like both are on the left side or both are on the right side. Contralateral is opposite side. Okay, so if we take a look at this picture, the spleen is behind your stomach, helps you store blood, and it's part of your immune system. The spleen is here and the gallbladder is right here. So they are on opposite side, right? Contralateral. Contralateral. What about stomach and the liver? Okay, stomach and liver are stomach is right here. Liver is right here, still on opposite side. So also contralateral. But liver and gallbladder, they would be ipsilateral. They are on the same side. Next, we have deep and superficial. Right? Deep is closer to the core of the body. Superficial is, closer to the surface of the body. So bones are generally located deeper than muscle. The muscle is on top of the bones, right? Skin to muscle, what do you think? Skin to muscle, what's going on?
Superficial?
Superficial, yes. Nothing is more superficial than your skin. Maybe the hair on the skin, but like other than that, skin is pretty superficial. Brain is something, the cranium. The cranium is your skull, okay? Protects the brain. So the brain is deeper. Deeper than the cranium. The pericardium, I mean, you don't have to know what it is because the question tells you what it is. The pericardium is a membrane that surrounds the heart. We can say the pericardium is, because it's surrounding the heart, right? It's outside of the heart, so it's superficial to the heart. All right. Next, we're gonna talk about some planes. Over here. All right, we can cut the body using a plane. Okay, so you can cut it this way. Okay, this would be called a sagittal plane. Sagittal plane, which is what this one is. Okay. This one right here, the pink one I'm coloring, that is a sagittal plane. Right there. Okay. Look at me for a second. If you can cut, you can use sagittal plane to cut like anywhere. Like you can cut it here, 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 right? But if you cut right down the middle, right down the midline, that's a mid-sagittal plane. Okay, so this one is mid-sagittal plane. But remember, you can actually cut like anywhere. It basically, the sagittal plane divides the body into left, right. Okay. It's only mid-sagittal if you go down the center. Okay. So for example, like these are CT scans, right? These are mid-sagittal view of the body. You have to know what views you're looking at, right? All right. What about this one? This one, we are cutting it into top and bottom. This is transverse plane. divides the body into superior and inferior. Okay, so cut like this way, parallel to the floor. Okay, cutting like this, the sagittal is parallel to the walls on your side. And then finally, we can also cut this way. Can also cut this way. This is called the frontal or coronal plane, which divides the body into anterior / posterior. Okay, so cutting like this. Into front and back. All right. So here is the definition, which you just wrote down. And this is just to show you like actual pictures of what it looks like if you cut at different locations. Okay. This is all the stuff that's inside the abdomen. If you cut a transverse slice, you can see things like the liver and you know, part of the stomach and so on and so forth. So like we're gonna look at a lot of diagrams next semester, and when I show you a picture, you have to know which direction you're looking at it from, right? Are you looking at it from the side, from the top? Because depending on which view you're looking at, the labels might be a little bit different, right? Okay, let's take a look at this. Select all the correct answers for the blank. The right thumb is blank to the left big toe. Choose all the right ones. Here, I'll launch the poll. See if you feel like doing it today. Okay, one person answered, and I think they're correct. Okay, so the right thumb is superior. It's definitely, the thumb is definitely higher up than your toes, right? If it's superior, then it cannot be inferior, right? Thumbs are, like if you in anatomical position, the thumbs are further away from the middle than your toes are, so they are lateral. Okay. Thumb is not really in front of the big toe. If anything, it's behind it. And the right side and the left side, contralateral. Contralateral. On the opposite side, right? Which of the following best describe the plane used to cut a salmon stick? What do you think? Which cut is this? Think of the fish. Think of the fish. Salmon stick.
E.
B, sagittal. Okay. And then someone said coronal. Yeah, this is coronal. Okay. Like if you think about the fish, this is anterior and this is posterior, right? So if you cut the fish this way, then you get yourself the salmon steak. Okay. If you're filleting it, okay, you're filleting it, that would be sagittal. Okay. I have never seen anyone cut the fish using transverse though. That yeah, that wouldn't make any sense. Okay, I'm gonna stop here today because I know you have that, you know, super long quiz of mine to do. We will finish up lecture 10 on Wednesday. We will start a little bit of review. No class next Monday. Sorry, no class next Tuesday. Okay, you guys can just, I'll post some review material for you to do, but you will just study for the final test. And the final test will of course happens on the 13th. Okay. So no, on the 14th. Sorry, I keep on mixing it. So just to clear again, I'll see you again on Thursday to finish up the lesson and do a little bit of review. And then no class next Tuesday. Okay, you can just study on your own, watch the review material. And then final test is going to be on Thursday next week. Okay, the 14th. All right, good luck with the test. Okay, take advantage of the 2 attempts. I will see you on Thursday. Have a good one, guys. Bye.